Our next speaker is Professor Rikky Muller, an assistant professor in ECS, co-director of the Berkeley Wireless Research Center, and a core member of the Center for Neural Engineering and Prosthesis, which we heard about already a little bit from our first speaker, Jose Carmena. She's also an investigator in the Chan Zuckerberg Biohub. Her research group focuses on emerging implantable medical devices, developing low-power wireless, microelectronic, and integrated systems for neurological applications. Professor Muller is also co-founder of Cortera Neurotechnologies, Inc., a medical device company founded in 2013 that is commercializing a neural implant device and has released a family of products for animal neuroscience research market. At Cortera, she previously held positions as both CEO and CTO. Her talk today is on advances in neuromodulation technology, which will enable patient-specific therapies that will result in improved outcomes and reduced side effects. Welcome, Professor Muller.
It's backwards, that goes forward.
Great. Thank you. Thank you so much for the introduction. Thank you all for being here today. So I'm going to talk to you today about a class of devices known as neurostimulators. And these devices provide pulses of current as therapy. They interact directly with the brain or with peripheral nerves in the body. And this is opposed to pharmaceutical medication, which is nonspecific, systemic, doesn't act very quickly, as you all know, has a lot of side effects. And electrical currents can be extremely targeted, very fast acting and personalized. And so you might be thinking, wow, why aren't we all doing that today? Well, okay. Well, the devices today require surgery to implant. And so you would really only have one of these devices implanted if pharmaceutical medications were no longer working at all. And so this is a deep brain stimulator. It's largely used today to treat the symptoms of motor disorders, such as Parkinson's disease, essential tremor, dystonia. There are 200,000 people in the world walking around today with deep brain stimulators. And essentially what they are is you can think of them as like a pacemaker for the brain. In fact, they are actually repurposed pacemaker technology. You can see that all of the electronics here in the chest cavity. That's where the battery and the pulses of current come from. And then there are wires that are run subcutaneously up to the top of the skull where burr holes are drilled. And very long electrodes are placed in deep structures of the brain. We're talking about 7 to 10 centimeters long. So now you're thinking, who would actually sign up for this? Well, let me show you. Actually, I'm going to let Andrew show you. This is Andrew Johnson. He's 39 years old. He has Parkinson's disease. And here he's demonstrating the symptoms of his disorder, known as bradykinesia. He has stiffness, rigidity, difficulty speaking, difficulty initiating movements. You see this sort of classic tremor that we usually associate with Parkinson's disease. He explains that if he tries to stand up, he'll fall. And he is implanted with a deep brain stimulator, as you see in the center. And when he turns the device on, he almost completely regains motor function. Now, these are not random movements. These are actually the tests he does in the doctor's office to demonstrate dexterity. He speaks very clearly. I know you can't hear him right now. But if you met him or if he were in the audience, you would not know at all that he had Parkinson's disease or that he was implanted with a deep brain stimulator. So clearly this technology is completely transforming lives already today. But there's a process before this can happen. This is the best deep brain stimulator money can buy in the world today. This was on the cover of the Journal of Neurosurgery just a few months ago. This is what's being used by my colleagues at UCSF. And it has some limitations. Okay, it's kind of a crude device. It has just a few electrodes, a few channels that have to be very, very precisely implanted with a significant amount of imaging that goes on in the OR. And then once it's implanted, the patient goes home, recovers from the surgery, comes back to the doctor's office. The doctor runs tests, they tune the parameters, send you home, come back, repeat this every couple of months. And to get the parameters just right for a patient can actually take up to 2 years. And for some patients, it's hard to ever get it right. So clearly, being able to sense what's going on in the brain and have a device that operates in closed loop and can automate finding the optimal stimulation parameters for a patient would completely change the landscape. It would allow much more rapid outcomes and much more accessibility because patients don't constantly have to come back to the hospital. But there's a lot, so we actually set out to design a much more intelligent device than exists today. And we weren't limiting ourselves to one specific disorder. We actually wanted to target a wide variety of disorders, and disorders that were very complex, whose symptoms weren't constantly active. So for example, neuropsychiatric disease, major depressive disorder, post-traumatic stress disorder. So disorders that manifest at the systems level in the brain, whose symptoms are not always active. And we needed a much more highly scaled, much more intelligent device. We needed a device that could sense from multiple regions of the brain and do broad-based sensing, could perform computations on what it was sensing. So it's looking for specific biomarkers of disease, so specific indications of things that are going on. And then it would be able to provide targeted therapeutic stimulation and figure out what's the best way to stimulate, where is the best place to stimulate in order to alleviate these symptoms. So technologically, the key to having this kind of platform device that had so much capability and so much computation that would be allowed is that we had to design our own custom electronics in order to see this kind of device. And so I'll talk about that in just a moment. That's sort of at the heart of what we do. But just so you have a sense of why else is this closed loop sensing really important? So this is an example. This is actually a commercialized device that is, what they're doing is they're sensing exactly when a seizure is happening or about to happen. And then they provide stimulation only at the time that the seizure is actually occurring, rather than all the time. What does this do? This saves battery life, first and foremost. This is important because that battery is surgically replaced. Okay? Also, it alleviates side effects. I mean, if you're having one seizure a day, while that's extremely disruptive to your life, you really don't need a device on all the time. You need it on for that 1 minute every day. So this company went forward and has a very successful device that is commercialized for this purpose. And what they're doing is they're sensing some activity in the brain. And when they detect a seizure, they turn on the stimulation. And eventually they turn it off, and there's a period of recovery. When they turn on the stimulation, the thing to note is that you really can't do any sensing. They actually turn off the sensing while they're stimulating. So you can't monitor what's actually happening when you're delivering the therapy. Well, why is that? This is what it looks like. This data is actually from our neurosurgical colleagues at UCSF, Dr. Philip Starr. And this is from a device implanted in a patient with Parkinson's disease. And when the stimulation is turned on, that's all you can see. You can no longer see those tiny little brain signals that we're looking for. All you see is this huge artifact. And it's spread throughout the time domain, throughout the frequency domain. It's just hard to extract any meaningful information out of it. So a lot of people we'll just turn off the recording entirely. Well, we decided, that's not good enough. We really need to see what's going on while we're delivering the therapy. We need to know exactly how to adjust it. And we actually took a hardware-based approach to this problem, where we saw, okay, we're delivering these pulses of current, and those are causing these big artifacts and deviation, but those pulses are really short. There's no reason we can't recover right away. And what's happening actually in these systems is just limitations on instrumentation that is not really designed well to handle these huge interferers. And so we decided, well, we can design better circuits than that. And so we did. We designed circuits that took these really long, persistent artifacts and adjusted them so that you only saw a perturbation exactly at the point where you stimulate. These are our chips that we designed in order to do that. And we had 64 channels of simultaneous recording and stimulation. And these chips could very, very rapidly communicate with the computation hub so that we could aggregate all the signals and send commands back in order to stimulate. They're very low power, they're very tiny. So what you see here are those chips placed in a ceramic hermetic package that is safe for human implantation. So it's extremely small as well. And of course, before a device can ever go into a human being, we actually test them out in animal models. And so we teamed up with Jan Rabai and Jose Carmena, and we built this platform in order to test this device in non-human primates. And so what we did is we took 2 of our chips, and for a total of 128 channels, we put them on this little board. It's about 1.5 inches by 1.5 inches. On the other side of the board, we put everything else. We put a microcontroller FPGA so we could do all that computation in closed loop in real time. We put a bidirectional radio so that we could talk to it completely wirelessly. Of course, we have all kinds of other peripheral things, power regulation, etc. And we implanted them in primates that are in Jose Carmena's lab. And the very first thing we did is we just wanted to make sure, okay, we can record from these devices, and what we're recording is neural, and it's all good signal quality, and we can record it over long periods of time. And then, just to make sure that we had designed our circuits correctly, we did this little experiment where we weren't stimulating, and then we turned on our stimulation. And in fact, you see very, very short perturbations that are happening. We can actually, on our device, completely take out those artifacts. And now, while we're stimulating, we have the complete underlying neural signal. So now we can do true closed loop, real time, and that's the signal that we're closing the loop on. And when we turn off the stimulation, you really can't tell the difference. It's really essentially the same signal. Of course, we've fully quantified that. So these primates, they're trained to perform a task. The task they're performed to train is they have a joystick, and there's a cursor in the center of the screen. And then a peripheral target appears. They get a go cue. They move that cursor to the peripheral target, and then they get a reward, as Jose was talking about earlier today. Turns out monkeys don't work for free. So we actually have them repeat this task hundreds of times. And this allows us to take the data to train our algorithms offline. And so what we do is we look for very specific neural signatures of motor planning, motor execution, and completion and reward. And the way that we do that is we're looking at very specific frequency bands known as the beta band and the high gamma band. And you can see that here, the primate is in motion, and you can see these features modulating very clearly. And so we use that in a closed loop context where essentially what we're doing, now this is done totally blind autonomously entirely by the device. The device is recording the neural activity. It's sensing when the primate is about to execute its task, so when it's planning the motion. And then we stimulate in order to disrupt that motion. And so what that allows us to do is to bias the reaction time of the primate. And this is, honestly, this is not a new neuroscientific result. This is actually a known neuroscientific result. But this is the very first time it's ever been done autonomously in closed loop based on the neural state, the recorded neural state of an animal. So now we have this platform device that we can actually program in all kinds of clinical contexts or research contexts and use in closed loop for the first time. And so we have a number of different opportunities. We can now monitor patients during treatment, which we couldn't do before at all. We can facilitate biomarker discovery, so we can watch as symptoms are happening and figure out, okay, what is the neurological signature that is associated with that side effect? And then we can come out with strategies of how to mitigate that. We can deliver therapies on demand exactly at the time that's needed. We can automate finding optimal therapeutic parameters, as I was describing earlier in the talk. We can map connectivity in new ways. So this is actually pretty exciting, because for the first time, you can think of this as we can now ping the network and immediately measure the response, which is something that can't be done with current instrumentation. And we can study how stimulation affects changes in the brain over time as well, since we're collecting data continuously. And finally, we can explore ways to, instead of training these algorithms entirely offline, we can actually give these devices the ability to learn. And that's one thing we're working on in my lab. We actually made a bunch of these, what we call wand minis, or little tiny versions of this device. It's about 1 inch by 1 inch, so that we could deploy them more widely, try them out in various disease contexts. So one of the things we're working on is incorporating machine learning for seizure detection. And what we've been finding is that actually the signatures of the seizures, so the features of the seizures that happen, they actually change over time. They're not really a static set. And so one thing that we can do is we can take our seizure data, take a certain amount, we train our model offline, but then as we go, we have online learning algorithms that actually update the model over time. And that allows us to actually maintain the sensitivity of our detection for a longer period of time, rather than just training offline with very well-known detection algorithms, which sort of lose their sensitivity over time. So we're also applying this technology to peripheral nerve stimulation. So this is very similar to deep brain stimulation technology. Again, it's current pulses that are applied directly to peripheral nerves. Today, this is commercialized to control seizures in patients with epilepsy, but it's actually being investigated for a wide variety of clinical applications. So for control of inflammatory disease, for control of respiratory disease, asthma. So for control of incontinence. There's a huge variety of clinical applications actually for this technology. What you can see is that it's just as invasive of a device as the deep brain stimulator. But ultimately, what we want to go for, now that we're not drilling holes in the brain, can we make really tiny little devices that don't have to be implanted surgically at all? We can implant them laparoscopically or through a cannula, or best case scenario, we can inject them into the human body. But for that, we have to make them extraordinarily small. But they still have to do all the same functions as this really large device. So how do we do that? So one thing we've been looking at Berkeley for an extremely long time is coupling power and data into really tiny little devices, say about 1 millimeter cubed. And it's really hard to couple a lot of power into tiny little devices using electromagnetic waves. I'll explain why in a moment, but one invention that Berkeley professors came up with, so this was a collaboration between the Maharbis, Carmena, Rabai, and Alon labs, is that they recognized that using ultrasound, which propagates very efficiently in the body, and having ultrasound transducers of about the same size that are collecting the power from the ultrasound, now we can actually, collect very useful amounts of power and do something real with that, make high-performance devices. And so why is that? Okay, tissue attenuation. EM fields dissipate very rapidly into tissue. Ultrasound propagates extremely well. The FDA intensity limits are actually a lot higher on ultrasound because it doesn't heat the tissue in the same way. It can be focused, just like RF fields. And actually, most importantly, the wavelengths are extremely small. That means that it allows us to resonate devices that are at the millimeter scale or below the millimeter scale. So this was the first demonstration, known as neural dust, that the Maharbis, Carmena, and Alon groups did. What they basically showed was that they could beam a pulse of ultrasound into a tiny little device and get a reflection back and be able to read out some biological signals. So beautiful little demonstration. Here was the device. It had the ultrasound transducer, and I'm going to call it an amplifier, but it's really just a single transistor there in shunt with the device that's picking up some signals. Very, very nice first demonstration. What we've done since, and actually since I joined the faculty, was we want to bring some of that higher performance neurostimulation and neural recording into this tiny, small scale. And so the first device that I collaborated with them on and worked on, we called StimDust. So it's a tiny little peripheral nerve stimulator. You can see it here on a fingertip. The whole volume is about 2.2 millimeters cubed, and it's extremely efficient. This is so important because I said, okay, we can couple hundreds of microwatts in. We need all that power to go to the nerve. We can't waste it on electronics. And of course, we need this to be very precise. We need to be able to communicate with it externally, so we need these little tiny custom chips in order to do all that. And the key to how we dealt with this efficiency is that, yes, this is a wireless device, but it's not a generic one. We're not running Bluetooth. We're not running Wi-Fi. We don't have to send megabits of data. We're doing something extremely specific. We're telling it when and how to stimulate, and that's all. And so we encode that just in the waveform shape of the ultrasound that we send in. And that allows us to do this without memory, without a precise clock, and we can get very, very low powers. Our entire chip consumes 4 microwatts. So here it is, and it's actually implanted on the sciatic nerve of a rodent. And what the sciatic nerve does is it controls hind leg muscle contraction. So the leg of the animal is actually kicking as we stimulate it. So we had this test set up where we communicated with these devices wirelessly through the ultrasound. We gradually increased the amplitude. And what we could do is we could monitor the force of the hind leg muscle. And as we did that, we saw that as we increased the amplitude, we could actually get full-scale activation of the nerve from 0 all the way to full kicking. Of course, we want to be able to record from peripheral nerves as well. And the way recording works, as I briefly mentioned earlier, is that we send a pulse of ultrasound. Now, of course, we have to get information back. So what we do is we wait for the pulse to come back at us. What encodes the information? Well, we send that pulse to the implant. The implant powers up and it modulates that pulse in some way. And that's what we collect back at the external device. So we have this vision to have a whole bunch of these implants all over the tissue. And so this creates a certain amount of complexity because ultrasound takes a certain amount of time to propagate into tissue. And so if your implant is far away, you have a lot of time. You can send a long pulse, you could send a long bit stream back, but if your implant is very close, you don't have time, right? That pulse has to be very short. It gets there really fast, it comes back really fast. Now we don't have time to send a long bit stream. And so the way we got around this is we decided to send everything in analog instead of long digital bit streams. That allows us to use very, very short pulses. So it kind of acts like an AM radio. One challenge was that actually the reflection coming back was not linear. And in order to encode in the amplitude, you need something nice and linear. So I have clever students that came up with really interesting circuits and figured out how to linearize this reflection so that we could send everything on an AM wave. And that's exactly what we did. Here's the device. We actually got it down, in this case, to 0.8 millimeters cubed, so we're at the micron scale, finally. It has a teeny tiny chiplet that you see here, and just the piezo that we always had. Now, we're working very hard to shrink these even further, because like I said, we want to be able to scatter them all throughout the tissue. So, just to show you that we are actually able to read out more than one at the same time. And this is the way that we do that. I said we encode in the amplitude, but we're also sending them on different subcarrier frequencies. That way, when we collect them back and we down-modulate the signal, we can tell them apart. And so this, what you're seeing here, is we have two motes that are sending a 100 hertz sine wave and a 500 hertz sine wave. And this is the pulse we get back from the combined signal. And we're still able to tell apart what came from A and what came from B. So what's great about these is they're minimally invasive. We can spread them with wide spatial coverage, but at the same time, high resolution. And we have a vision of having a network of these with simultaneous operation and have them perform both recording and stimulation and ultimately be able to use these devices in closed loop. And looking into the future, as we're shrinking these down into the micron scale, we can start thinking about actually putting them in the brain. All right. I want to acknowledge my awesome research group, half of which actually works on optical interfaces to the brain and body, and so that's another talk for another day. And of course, my various collaborators of the work that I presented, Jose Carmena, Mike Jordan, Michelle Maharbiz, and Jan Rabai, and I acknowledge our funding sources as well. Be happy to take any questions.
We have a question in the back there.
So, great talk. So when you have a number of these small things that are embedded into the body, are you worried that they might move? And if so, how do you locate them inside it?
Yeah. So we can use this. So for location, we can use the same mechanism of the readout. So we're looking at reflections. So you can also effectively image their location that way, the same way you would with ultrasound. In terms of movement, it is true that if you just inject a device into tissue, it's likely to migrate, but there are a lot of surgical techniques that we're using to actually affix it. So what was hard to see, or what I didn't show details of, is that our peripheral nerve stimulator is actually cuffed all the way around the nerve. And there are different ways of suturing these devices as well. And depending on where they're implanted, compression can do the same thing. Great question.
I'd like to ask a question. So the first part of your talk, you were showing us a direct neural electrical interface.
Yeah.
And in Jose's talk, he talked also about the bed of nails array. That's different than the one that you're using.
Right.
He told us that was the only clinically approved implant. So could you talk a little bit about the comparison and sort of where these implants that you're talking about,
Yeah.
how they connect, and can they connect to the work that he's doing?
Yeah, excellent question. Okay, these are kind of subtle points. I did show electrode arrays that are clinically approved for human use. Okay, what Jose was talking about this morning was something very specific. It's an array for essentially for brain-machine interfaces. So what you need are microelectrode arrays that depress into the motor cortex, which is close to the surface of the brain. So there are other arrays that sit on top of the surface, but you're not going to get the resolution that you need to perform the motor tasks that Jose was describing. What I was showing was a combination of those surface arrays, which are very commonly used in humans, both intraoperatively and for implanted devices, but also the deep brain stimulation probes, which are also approved for use. But the surface arrays and the deep brain stimulation probes, those don't work well for brain-machine interfaces because they don't really interact with the motor cortex in the same way.
Hi, I'm curious to know which therapeutic opportunities you think are most exciting if you implant multiple devices within tissue.
Yeah. Are you talking about the peripheral nerve, or are you talking about the brain devices?
Well, I mean, I guess in the last example.
Yeah, in the last example.
Yeah.
Okay. Yeah. That's an excellent question. I think, the sky's the limit. I was, specifically the experiments that we had done showed the interaction of the nerves with muscles. So, for example, if you think about connecting up an active prosthetic to the peripheral nerves in the remaining limb, now you can think about having direct control through the nervous system, where you have a whole bunch of these implants that are then, essentially connecting to the external device. So that's one, in the future kind of application where we can have bidirectional communication of a prosthetic device with the nervous system, and that's kind of the holy grail of prosthetics.